I have the pleasure of introducing our next speaker. As a science-based organization, the OFAH is always seeking to have the best available research and data to inform fish and wildlife management decisions. Our next presenter is using a relatively new tool in the Fish and Wildlife Management Toolbox, DNA. Dr. Brad White is the director of the Natural Resources DNA Profiling and Forensic Center, as well as professor of biology at Trent University. He holds a BSc in botany from Nottingham University and a PhD from McMaster University. Dr. White has been at the leading edge of his field throughout his career, establishing the DNA Diagnostic Laboratory at Kingston General Hospital, the first of its kind in Ontario. He also created the Wildlife DNA Forensic Laboratory at Queen's University, before spending time at McMaster University and the University of Kent. In 1997, he moved the laboratory to Trent University, and in 2001, formed the Natural Resources DNA Profiling and Forensic Center, as a partnership between Trent University and the Ministry of Natural Resources and Forestry. Dr. White has published over 240 peer-reviewed papers and trained over 60 graduates and postdoctoral students, many of whom are now faculty members at other universities in Canada and abroad. Dr. White is with us today to tell us more about his work using DNA in fish and wildlife management and enforcement. Please join me in welcoming Dr. Brad White.
Okay, I'm gonna wander around a bit. I'm a bit throat and tired. I had a 6 to 9 o'clock lecture last night, and I'm still recovering from it. I'm gonna have a couple of different themes going through this talk, and what that might do is produce a tangle or an interesting talk. If it's a tangle, I apologize. If it's tweaks your interest, I'm happy. So it's going to be a talk on DNA, and it's going to have some history. As you can see, I'm old, and I've been through all of the changes that have happened in DNA technology, unfortunately from the 70s. When I say 70s to my students, I think it's something like prehistoric times. So at Trent, we have a center. It was started with the partnership of the Ministry of Natural Resources before they became forestry. My paycheck comes from Trent and not the ministry. They're both coming from taxpayers, but from different sources. And so I want to show you some of the things that we do, but in the context of what is going to happen to the world, and why I think it relates to the previous talk of Angelo. If you can see this, it's a bit of a tongue-in-cheek app, but it's not far off. So here the DNA sequence, you can see there. So DNA sequence have those 4 bases, T, C, G, and A. And the idea in the near future is that you will have your DNA sequence in your iPhone, and the next time you want to pick someone up at the bar, you show each other your iPhones and tap them together, and either a green light comes on for compatibility or a red one for incompatibility. This is not far away. And already in some communities, as was said, I did run the DNA Diagnostic Lab at Kingston General. Some communities, Jewish community, requires you to procreate, have kids, and there are a number of serious diseases, genetic diseases, Tay-Sachs, and you are tested for Tay-Sachs, and the rabbi advises you your situation and whether you should actually get married or not, or whatever. This is coming very fast, and that's the one theme I want to sort of put into the talk, because it's related to wildlife conservation, and I hope I can convince you of that as we go along. So this is DNA. You see on the cornflakes boxes, and you hate it, the DNA double helix. Every rung of the ladder is a base, and there are a couple of billions in the talk, so DNA is at least 1 billion, if not 2 billion years old on the planet. And we are all linked to every other life form, because everything says the life as we know it only occurred once on this planet. And so every life form came from that particular event. It's certainly a lot better than Microsoft in terms of an information technology, and it allows both replication and mistakes. The mistakes are important, otherwise we'd all look like little bacteria 2 billion years ago. So the mistakes really are the important part of it. So we're going to be talking about a number of different species, and the important thing with the DNA is that some parts of that DNA are specific for a species, whether it's a particular trout, particular bird, moose, or whatever. And some parts are specific to a population, so we can tell, did it come from northwestern Ontario or eastern Ontario? And some bits are specific to the individual. So we're going to be using DNA variation in that molecule to answer a variety of questions. The closer two species are to each other, the closer the DNA sequence. So we share about 99% with chimpanzees, for instance. Some people might say 100% for some people. I want to sort of give you our framework for conservation and separate it from preservation. We're not trying to preserve what is. We're trying to conserve the ability to change as the world changes. So as climate change kicks in, as we screw up various parts of our wonderful province, how do species react to those changes and adapt to them? And that's really what DNA is all about. We do not want a field of identical Holstein cattle out there. So we want a lot of variability. So this is a picture of Rick Stankiewicz, who was a conservation officer that's worked with us a lot in the past. Rick seemed to retire a lot younger than I did. And these kids are kids at Trent who take a summer course in forensics, and both the OPP and the MNRF are involved with teaching these kids. So I want to touch as we go on. We're a university, we educate high school kids as well as university kids, and it's very important to think of this sort of passage of time in the future. And you can link it to DNA, that passage of time from 3 billion years ago to now. That molecule has lasted all that time. It's changed, obviously, but it's got that thread that links. Nelson is very important in this story, and like a lot of stories, was not well recognized. Back in the late 80s, Nelson came to me when I was at Queen's and said, this DNA stuff can do stuff for conservation officers. And he asked me to speak to the CO organization that year. And Nelson actually produced the first DNA case in Ontario that was finally presented in court. And Nelson unfortunately died in the 90s, but sorry, not in the 90s, in the 2000s, just before a building that really, this is the building we work in, and I really dedicate the building to Nelson, because he was the kind of little seed that started everything going, including the forensic programs at Trent. Without the Wildlife Forensic Lab, there'd be no degree program. Fleming wouldn't have its forensic program. It all comes from that idea that Nelson had way back there. So this building, we're in there. Ministry of Natural Resources and Forestry Research, Fish and Wildlife are in there. And it's been the home of a lot of activities since 2006. Now this is personal, and I think you'll understand sort of why as we go through it. So this is my son-in-law. He was OFAH member and was absolutely dedicated to hunting and fishing. He died suddenly 4 years ago of leukemia. And I can get into some of the DNA aspects of that, but I won't. So Kirk was very much a Peterborough person, even though he came from Barrie. He got his degree from Trent. He was an MNR employee. He played for one of the MNR hockey teams, and they were just a little bit better than the Maple Leafs. And you'll see why that sort of theme comes. So this was my main contact with this organization, hunting, sort of fishing, and all of the seasonality we knew because Kirk would be getting ready to do his thing, whatever species it was. This is my granddaughter, his daughter. She's now a hockey player, and she had to pick it up, not from her dad, because her dad had died. And that was important, I think, from what Angelo said as well. How do you pass on these cultures when someone dies? And so both OFAH and MNRF were very kind to the family, actually. And this is Brenna. Brenna was looked after a lot by both of the organizations, and Kirk actually has a memorial fishing derby every year that's dedicated to him and the girls. So these organizations are bigger than just all the things Angelo said this morning. I think they're really family groups. They are the village that can help educate the future generations there. The other interesting thing is that Brenna's got a quarter of my DNA, got half of Kirk's DNA, and that sort of is amusing because Kirk and I, as father-in-law, son-in-law, had that normal kind of relationship, and then imagining that our DNA is intertwined now in that individual, this is kind of, I'm sure, puts a smile on his face. Okay, so I'm going to get into the wildlife, but just give you this backdrop. In 2001, our species was sequenced, 3 billion base pairs, cost $3 billion in the public sector. The private sector claims it only cost them $300,000. So we can, but it's a lot of money. Now it costs $1,000. Each of you will be sequenced probably before you're dead, and it will be applied to managing your clinical situations. If you do get cancer, it will be used to actually design the amounts and drugs, the chemotherapeutic drugs, for instance. And so there's a lot more careers coming on that will follow from you having been sequenced. So we talk about next generation sequencing. These are the machines that can do it for these kind of prices. They now sequence everything. So you'll see outside there's a project on the moose genome. We've just sequenced a whale genome. So these things are coming off the sort of laboratory benches rapidly, and they have a lot of power in the tools that they reveal. So you'll see this a lot, and you'll be dealing with it in a number of different ways, as I said, from personalized drugs to making reproductive decisions. And you can see why we have a certain advantage being north of the 49th parallel in that our healthcare is not dependent on your genetics. It's the healthcare people get hold of it, you might finish up with a high premium because you're genetically not in good shape, for instance. So if you've got high risk of heart disease, cancer, and things. So this is powerful public policy stuff, and that's what I was doing last night with my class. We talk a lot about the ramifications of all these new technologies to public policy. So these are the tools, badgers or moose or Atlantic salmon, all sort of sequences out there that you can use for the kind of things I'm going to be talking about. So species are scattered across the landscape, and what I like to think about, instead of, when you're scared giving a talk, the advice is look at your audience and pretend they're naked or in their underwear, and then you won't be so scared. What I do is think of you all as DNA molecules just sitting there. And all of you actually have left DNA on the table if you were a forensic case, so we could actually find that you'd been in this room. So we look at DNA across the landscape in a conservation sense, as well as enforcing the statutes for the conservation. So I'm going to talk about wildlife forensics and some of the work that is being done on the biology of species. Now, I've spent my life trying to publish papers in good journals. My mother never understood me. I had an article in Reader's Digest, and she suddenly thought her son must be doing something right. So there was we've had quite a few kind of more popular articles, but this one sort of dealt with the issues and the similar kind of work we would do with a police lab. The only difference is they only deal with one species, because we are the bad species, and we have to deal with everything else in the... These are some of the things that we would be dealing with. Kill sites, this one's a kill site on a road. And the issue is the same in a police investigation, trying to link the scene of a crime with some other evidence. Could be meat in a freezer, might be meat in a truck, might be blood on a snowmobile, ATV. So what we're trying to do is use those DNA sequences to try and identify that animal which has been killed illegally on this road to some other evidence that might be in a suspect. So those of you that can see, I couldn't see anything, but my eyesight's not what it used to be. There was a bunch of peaks, they're different colors. Those of you that follow CSI Miami or any of those things and hear profiles, that's what a profile is. This profile happens to be a moose. If it was a human, it wouldn't look a lot different. If you can see, there are twin peaks. It's not the noisy stuff next to the peak, but you'll see two blues next to each other on the left. You'll see a couple of greens, and then a couple of blacks. Each of those peaks represent a chromosome that came from the mother or father of that moose. So because you're diploid, you have two copies of every gene, one from mom and one from dad. When we do the profiling, we get two peaks. And so we get a profile from the blood on the road, and then we profile meat. And if you, again, look at the TV programs, the data that is presented in court is one of probability. So if you're accused of killing this animal on the road because the meat in your freezer matches it, same profile, one of the questions you need to ask is, well, how do I know that every moose out there doesn't have the same profile, right? So you need to know what's the probability of a second moose in Ontario or that management unit in that district having this profile. And so those probabilities are presented. With moose, the probabilities are not great. They're usually in the hundreds of thousands to millions, not bad. But with white-tailed deer, they're more in the millions, many millions to billions, just because white-tailed deer are much more genetically variable than moose, because moose came through the Ice Age and are not very variable. But sufficient power to present in court and allow the judge to draw the conclusion. So much of the work we do is on moose. We do quite a lot on deer, but a lot more really on moose these days. And I'm going to touch on some of the other species we work on, because it was mentioned about coyotes and wolves. And DNA has been used to unravel that. We've been putting DNA analyses on these critters now since the 90s. We started in Algonquin Park, blindly thinking the animal in Algonquin Park was a gray wolf, because everyone said it was a gray wolf, and I didn't know anything about wolves. When we looked at the DNA, it wasn't a gray wolf, and much more related to a coyote, actually. And so this is a stuffed critter that comes from Algonquin headquarters. And what we've been doing is historically looking at DNA, as well as DNA really across North America to understand what has gone on in this continent with respect to coyotes and wolves. And now we've got this situation with a lot of bigger eastern coyotes, especially in the Peterborough region, and having impact on farmers as well as some negative interactions. In Toronto, they quite like poodles and cats, right? So they're adapting to their new environment. There was not a single coyote in Ontario really prior to about 1919. So coyotes are a completely new visitor to this province. And if you follow the newspapers, you see they get bigger. They got bigger from 1919 to the 30s, and they headed east instead of west. They finally finished up in Newfoundland. They caught an ice floe. So there were 3 times these critters landed in Newfoundland, and the Newfoundland Natural Resources debated whether they should let them on or not, whether they had a passport or border guards, or they let them on. So now, not only do we have all the 150,000 moose that never used to be there, we have all of the eastern coyotes that never used to be there either. So a lot of the biology of this goes back to 50s and 60s, but really there's the 3 players. There's the western coyote that came into Ontario in the early part of the 20th century. There's a grey wolf, which actually came here 1 million years ago from Eurasia, and the eastern wolf in Algonquin Park, which is closely related to the western coyote. And there's probably 300 plus of those guys still in Algonquin. And then there's what we call Canis soup. So the rest of our province has hybrids. So the eastern coyote is a hybrid between an eastern wolf and a western coyote, with a bit of dog thrown in. And then there's a reasonable genetic eastern wolf in Algonquin. It's a bit mutty. It's got coyote in it, and it's got some grey wolf in it. You go a little bit north, and the Great Lakes wolf is a hybrid between the grey wolf and the eastern wolf. And then finally, you might bump into a real grey wolf. So these guys have been hybridizing and really covering this landscape. So, whoops. This is one possible distribution of these critters across. So the yellow is more western coyote now, and then the brown color is a hybrid, is the Great Lakes wolf, and there's a different color just below that which hasn't come out well, which is the eastern coyote, which is essentially colonized all of the forest that used to be eastern wolf. So that's why I talk about change happens. You can imagine those first dumb Brits that got off the boat in the 17th century. They'd killed their wolves sort of back in the 12th century. Suddenly they want to be farmers, and now there's a whole bunch of wolves again. So immediately they're killing wolves, they're cutting down trees, the whole of the North American East landscape changes. That wolf is basically eliminated. I call it the Loyalist Wolf because I think the guys in Algonquin are the ones that crept up from the US when they were being persecuted down there. So they sort of came over with Kingston Loyalists at some point there. But this landscape is very much a landscape of human colonization in North America. And now we've got a new DNA across the landscape, and you can have different feelings about the eastern coyote, but it's been pretty successful. It was born probably around 1920. That's when the western coyote came in, hybridized, and swept right across eastern Canada and then south in the US. So that's what I'm saying. Change happens, and it should be celebrated because we will not manage the climate and we will not manage disease, and we need our species to respond to all these changes. So again, you can't see it, but this idea of landscape genetics, so when you look at a map full of lakes, roads, rivers, I look at the same area and I look at DNA molecules. And the reason DNA molecules are important is we're all going to be dead pretty soon. some of us quicker than others. And the only thing that goes on are your DNA molecules. So the DNA molecules in specific areas are what's going to be important for the conservation of wildlife in those areas. So that's why I have this bias that DNA is the most important thing, because that's what we are conserving for future generations. So this is our lab. I'm still OMNR rather than MNRF on a lot of these slides. But we started working with MNRF in '88 when Nelson started it. And it sort of tracked around the province with me between Queens, Mac, and Trent. And we presented the first evidence on a DNA wildlife case in North America. And that was at the time of Paul Bernardo and the Scarborough rapes. And the OPP were pretty cheesed off when we were in court. And I was giving DNA on a moose poaching case, and they couldn't get DNA on a rape case because the Toronto lab for the Center for Forensic Science was so backlogged at that time. So we've been providing mainly for Ontario, but also for Environment Canada, Parks Canada, lots of the provinces and territories, depending on what capacity those areas has had at different times. And I can't even read it now, but okay, so sometimes we get into murders, very rarely. That one, I think, was a cat. We've got into a murder where someone left a bag of leaves with a body, and we were asked by the OPP to profile the leaves to find out if they were consistent with the shrubs in the suspect's house, right? So that we'll do that with cats or leaves or dogs for that kind of homicide. Other than that, we're essentially non-human. So these are the kind of things that our COs ask us: species ID, sex of the individual for enforcement of tags. We do sausages a lot. Sausages, being a Brit, is one of my favorite foods, but unfortunately, once I found out what are in sausages, it sort of turns you off a bit. So my advice is always eat a pork sausage, but beware of spicy Italian or honey garlic. So we do parentage analysis to enforce captive breeding issues and populations, as I said. So a variety of different things. Our samples are everything: bits of clothing, knives, guns, blood on trucks, whatever, to get DNA. We do a lot of feces work these days, non-invasive collection of DNA, hair. We've just got 3 Kenyans actually now being trained in the lab. They are Kenyan Kenyan wildlife, and they're doing rhinoceros tracking and rhinoceros horn work. Canada, Baird gave Kenya $2 million to set their forensic lab up, and so they came over to be trained with our technicians. So, as I said, this is the most common analysis, individual identification linking 2 things. And those 2 things can be anything from kill sites to trucks to freezers. We've found in certain communities these have had sobering effects once individuals know that we can actually find out where the meat in their freezer came from with respect to a kill site. So there's a big deterrent effect, which I think is very important for conservation. The odd bit of poaching is not the issue. It's when there's a lot of animals taken and they finish up in Toronto as sausages, for instance. So 40 animals or so are suddenly sort of taken away from our environment. Polar bears, people in MNR like Marty Obard, for instance, and others have been taking samples from polar bears, and there's kind of a polar bear registry like the human DNA registry now, where you can actually get an individual profile, and if a fur crosses the border, you can see what animal it might have come from. So we've been doing some of that with Environment Canada, and there's a big rhinoceros one kept in South Africa. So that's the sort of parallels with the African conservation work. Similarly, you can get information about the population that fur might have come from. And so that slide just telling you not only individuals, but the kind of area regionally that an animal might have been poached in or that fur came from. So this is the sausage. They being the Kenyans, are very interested because there's about 40 different animals used for bushmeat in Kenya, and they get ground up. And so we have techniques to figure out how much of each animal is in there. So in general, we find it is either Canada goose, white-tailed deer, or moose mixed with pork or beef. And we can figure out those kind of mixtures from the DNA analyses. So this just sort of breakdown of the kind of requests and samples that we get, mostly individual ID, species ID for a variety of purposes, mixed meat sausage work, and sort of other population-y type studies. So there might be issues of fish coming from one lake or another. The one problem with fish is all the stocking that's gone on in the province, so that's mixed up the genetics, so sometimes that's not as easy. But you look for pieces of DNA that give you information on that level, kind of lake level, population level, not individual level. And I do quite a lot of work in Taiwan and Hong Kong on dolphins, and so we do some work on illegal harvests of minke whales, for instance, and other dolphins that are often labeled as fish meat in some of the markets. These are the kind of variable bits of DNA that give you species. So when you're looking for illegal commercialization of whales, you look at a piece of DNA to see what species it is. Some minke whales have been legal, but some minke whale populations are illegal. Right? So we're not just focused on sort of the Ontario scene, but the bigger scene out there. There's quite a lot of fraud that goes on, and this is a fraud with caviar. And so often bad high-value caviar is labeled low-value caviar. So we can monitor which species those caviar came from and which part of Russia, for instance, it might be coming into our country from. So that would be Environment Canada types of work. We get into the Asian medicine market, tiger products, for instance. And that really started with bear gallbladders. So Canada was exporting large amounts of bear gallbladders to Asia illegally, but the bear guides, hunter guides with the Americans in particular, were allowed to sell them. And they can sell up to $1,000 a gallbladder. So when I first did this, we did all the species ID and had a case just outside Algonquin Park. And the judge was giving a $1,000 fine per gallbladder. Unfortunately, half, or fortunately for this guy, half the gallbladders were from pigs. So he had fraudulently put pig gallbladders in there to make his money, and I saved him about, I think, $15,000, $30,000 because of it wasn't illegal to sell a pig gallbladder. So we've become quite expert in the penis trade, and because many of the penises come from our East Coast seal harvest, and they are often used to mask harvesting of endangered species, things like the Australian fur seal, for instance. So we identify species from these. Again, they can be $1,000 or so, usually ground over your soup to give you some benefit somewhere. This one, as I said, a cat murder type of thing. We take the hairs and we match them. And so just sort of to wind down, the typical kind of thing is this, when the COs have got some indication that there is suspects involved in killing in a illegal zone. And we get tissue samples. These can be, again, blood, hair, meat from the various sources, the kill site, or again from trucks, whatever is there, clothes, blood on clothes, and so on. And it all goes the same way. It gets converted quickly into DNA, and then we will profile it. And you'll those kind of peaks, and then the same kind of things that I said before. We tell whether it's male or female using DNA, and we get the profile. And the COs have a variety of tissue collection, things from tubes to bits of paper. We do hair a lot. It's tissue. And then you get a result. All the profiles on all of those samples were identical, and they were all from a bull male moose, and then you give a profile and it goes to court. And you get a verdict. So the verdicts can be up to quite a large amounts of money, $50,000 fines, and confiscation of boats and trucks, so it's quite big deterrents for some of these. The bottom one for some of the more northern individuals is quite a penalty, up to 10 years ban on hunting, for instance. The Environment Canada monitor a lot of the endangered species breeding programs, and so we get involved with proving that what they say is happening. So one case was eider ducks, and an individual, they're not quite eider ducks, but I couldn't get a nice eider duck picture. So, so that pair was supposed to have had a lot of offspring, and these were being used to harvest the eider. And so we went in, the same thing as I started with, what does the DNA say? Well, the DNA says they had 3. And so the DNA profiles of all the others excluded those 2 as being the parents. So there was a lot of animals being taken from the wild for that kind of commercialization. Usually federal cases, the verdicts can be jail time and a bit more severe than the provincial one, especially if migratory birds are involved. So we are involved with Marty Obard and the ministry with the black bear monitoring program. The advantage of using DNA is you identify individuals. So when you think there's more animals in an area or less animals in an area, that is better understood if you know the individuals that are in that area. Because sometimes you can get a change in habitat use that doesn't necessarily mean there's more or less animals. And so the DNA profiles are as good as a kind of ID photograph in terms of these individuals. So this work involves the MNR putting up bait stations and attracting in bears, getting bear hairs, which we profile. So we've profiled thousands of bear hairs a year for 9 years, and so we can actually detect offspring of bears in regions as well. So all those data then go back to the ministry to estimate the number of bears in different regions. So this is the kind of sardine trap that brings the bears in. So across the landscape, we have these DNA profiles of all these different species. They allow us to give these probability functions on different species having a certain profile, for instance, and help in giving that evidence in court. And also identify where things like bear gallbladders might be coming from in the province. So one of our challenges is always keeping data straight. No one likes to write things down. And so you get scribbly bits of writing from the field with the ink running or whatever. And so moving more to handheld devices to get the data in, so you're not worrying about that kind of situation all the time. Just to finish, we partnered with the MNR with tracking raccoon rabies when it came in the 90s. So we were profiling raccoons. We profiled something like 5,000 raccoons across North America. The worry was that raccoons were coming up from Florida. So they were hitchhiking in Florida and getting on a truck, and the disease was in Florida and would then come up to Ontario. So we were looking for migrant worker raccoons that would have come up, and they didn't seem to be able to cross the Niagara River. Maybe it was too steep, so we didn't see anything crossing there. Animals crossed the Thousand Islands, and that's where the sort of outbreak was. So these are the kind of things along the St. Lawrence, the outbreak. Then the MNR went in and we would be doing profiling, and with the Canadian Food Inspection Agency, we profiled the virus as well to see how the virus was traveling. Sensitive of the relationship between Ontario, Quebec, and New Brunswick. After we were infected, Quebec was infected, New Brunswick was infected. So who infected who? Fortunately, the US infected all of us, and it did not go east-west at all. It came south to north in each case. And you could do that tracking because of these variable bits on the virus. Oops. So this, I was changing jobs a while back. So we give these programs to the high school kids and the university kids. They get to take the samples. In this case, we kill one of them every week. So there's a murder, and then one of them is set up as a suspect. So they are profiled, and then the evidence is collected, and there's a defense and a prosecution and judge. So at the end of the week, they learn how to present this evidence. Angelo said, kids having fun. Well, you should see this crowd learning about blood splatter without knowing they're learning physics and all of the other math involved in figuring out, how did those blood spots get there. Learning chemistry with the DNA profiles and extracting DNA, learning statistics with the probabilities. So after a week, they've learned a heck of a lot more with a lot more fun than generally in the university type of classes. So this is sort of the wrap-up. Sequencing, next generation machines produce sequencing and profiles, have a big impact, I think, on the front line of conservation, which I see enforcement. There's no point in having our wildlife laws for conservation if they're not enforced, and these tools allow that kind of enforcement. We really haven't lost any cases since we started back in 1988. We've done, I think, better than most of the human labs. And we've got now, with many of the species, idea of the DNA across the landscape. As climate changes, things like the southern flying squirrels moving north, hybridizing with the northern flying squirrels, we're better to understand just what's happening as a result of that kind of change. And so just sort of this thread, which has been my life, just to understand it, that it really is what we give to the next generation. So all of the fish and wildlife that you enjoy is as a result of this thread being passed to the next generation, as well as you passing DNA on to the next generation, as well as passing on your cultural side of it, the hunting and the love of the wildlife, which obviously kids in Toronto don't get as much of. So I really think that the more of those kind of courses up in Peterborough, for instance, for Toronto kids, make it worthwhile. And we certainly get a lot of city kids. We get actually kids from all over Canada, Vancouver to Halifax, coming to our forensic camps to sort of show what our area is all about up here. So I think that will do me. So I hope it wasn't too tangled. My brain does go all over the place, but I hope that you kind of followed the wildlife with the DNA as well as the conservation. Thank you.
Thank you, Dr. White. As Robert said in his report this morning during our AGM, fishing and hunting is in our DNA, and now after this presentation this afternoon, we see how DNA can be used in fish and wildlife management as well as enforcement. So please join me again in thanking Dr. White for his informative presentation.